Hello, my name is Pamela Acker, and this is the long-awaited talk on avoiding the errors of scientism that was scheduled to be given at the Worcester Catholics Women's Conference in early October 2024. And it's now the Feast of the Holy Innocents in December of 2024, so I do appreciate your patience in the long wait that you've suffered to hear this talk. If you're familiar with my work from 2020, you may know that in 2019 I injured my ankle and never really quite recovered. And in 2024, just before the women's conference, I injured my other ankle. So now I'm a doubly not really very walkable. So I've been recovering from that, and I do appreciate your patience in waiting for this talk. So without further ado, let's get started with a prayer. In the name of the Father, and of the Son, and of the Holy Ghost. Amen. Saint Michael the Archangel, defend us in battle. Be our protection against the wickedness and snares of the devil. May God rebuke him, we humbly pray. And do thou, O Prince of the heavenly host, by the power of God, cast into hell Satan and all the evil spirits who prowl throughout the world, seeking the ruin of souls. Amen. Our Lady, Queen of the Angels, pray for us. In the name of the Father, and of the Son, and of the Holy Ghost. Amen. So when I was originally asked for a title for this talk, I gave it Avoiding the Errors of Scientism, but a couple of days later, a friend actually suggested a better title, which is Friends Don't Let Friends Trust the Science. So that's where we're going as we go about discussing this today. So it's a little bit of a roadmap of where we're going. And if you're not familiar with me, just a tiny little bit about my background. I worked at the Genome Sequencing Center at Washington University in St. Louis, not on the Human Genome Project, but I was there while they were finishing the Human Genome Project, and I worked on platypus and a couple of other smaller organisms. And then I also did some work in a novel vaccine vector platform at Vinigala Rao's lab at Catholic University of America, and I have an MS degree in biology instead of a PhD because I chose not to work with aborted fetal cells in his lab. So if you want to catch more of that story, there's a lot more I've given in other presentations, and I'll refer you to those. But just for now, I am a bona fide scientist. I've done some real research in laboratories, and I've taught a lot of people about science over a number of years. I've taught from elementary all the way up to college. So hopefully I will be reasonably able to articulate the problem of scientism as we're dealing with it here in 2024. So where we're going in this talk, we're going to talk a little bit about the nature of experimentation, so what it is and how it works, and we're going to talk about some of its limitations, and specifically with a focus on problems with medical research, because that's one of my areas of expertise. We're going to talk a little bit about the replicability of science and whether or not it's self-correcting. We're going to talk a little bit about the nature of scientists and dispel some of the myths regarding how objective they are compared to other human beings. And then we're actually going to get into scientism. So there's a lot of backstory here. We're going to define it. We're going to define some other isms that go along with it. And we're going to discuss the philosophical problems with those. So we're going to deal with practical problems first, and then we'll deal with some conceptual problems and philosophical problems. And even though the philosophical problems are more important, sometimes it just helps to get the practical out of the way first. And then we're going to talk a little bit about how I think we got here, and some of the ways that I think we can get out of this blind trust that we have in science as a method. So that's where we're headed. And let's get started with the nature of experimentation. So in our current cultural milieu, we're often tempted to think of science as something extremely objective. The science is the practice of science experimentation is very unbiased. The scientists themselves are only looking for the truth, and using these wonderful empirical methods that we've invented. They're always going to find it. But is this really the case? To take a look at that, we're going to look at a study conducted by Nate Bresnow, which was a very interesting experiment about experiments. So he and his team asked researchers to analyze the following hypothesis. Immigration reduces support for social policies among the public. So the idea that more immigration means there will be less support for social policies. And they asked 71 teams to come up with answers to that question using scientific methods. Now, this is, granted, it's a sociology question, But they're using the same kinds of empirical methods that we'd use in biology, and we'll discuss about how when we get into the harder sciences here in a second. But they asked 71 different teams to come up with an answer to this question. Does immigration reduce support for social policies among the public? And these teams came up with over 1,200 different models to answer that question. So let's let that sink in for a second. One question, 1,200 different answers. No two models were 100% identical. Now, every team settled on one or two models that they thought, they put forward as their final answer. And among these answers, about 60% said that the hypothesis should be rejected. That is, that immigration does not reduce support, but it increases support for social policies. About a quarter of them said that the hypothesis was supported, that immigration does in fact reduce support for social policies. And only about 13% of them said that the hypothesis was not testable given these data. Only about 13% of the researchers that were asked this question were willing to admit that they didn't know the answer based on the data that they were given. That's a really important point, and we'll probably come back and touch on that later. It takes a lot of courage to not just come up with an answer when you're presented a question. And if you've ever had children or you've ever taught children, you've probably experienced this pressure. When a child asks you why, you want to give them an answer. You don't want to say, I don't know. And scientists are no different than you in that situation. If they're looking for the answer to a research problem, they want to be able to say one way or the other, yes, my hypothesis is correct. No, my hypothesis is not correct. And there's a lot of pressure to do that. So it's very interesting that this experiment about experiments showed that there's a lot that's indefinite in experimentation. So what is an experiment anyway? We've been using this word. Let's talk about what it actually is. We can think of an experiment as an empirical test of a hypothesis. And you probably heard a hypothesis defined as an educated guess back when you learned the scientific method in elementary school. And so I'm going to ask you to discard that definition today because it's not a very good definition. A better definition is that a hypothesis is a testable prediction about the outcome of a situation. So in this case, they were testing or examining, they didn't actually test it, they did computer modeling, examining the hypothesis that immigration would reduce support for social policies. That hypothesis is testable because it's falsifiable. So I can say, no, it's not. So we're going to put a bookmark here. We're going to come back to the concept of falsifiability later. But it's an important concept when you're doing empirical science, when you're doing an experiment. It has to be falsifiable. If the hypothesis is not falsifiable, if you cannot say no, it's not science. So keep that in mind when people want to assert certain kinds of worldviews as though they were science. If you can't question it, if you can't say, actually, that might not be the case, if you can't do an experiment that would show, yes or no, then you're not really doing science. So hypotheses are good as far as they go, but the way that you phrase them can actually color your interpretation of the outcome. So when I was, shortly after I was working in the vaccine lab at Catholic U, I worked for a little bit of time in a lab that studied C. elegans, which is a tiny worm that's about 1 millimeter long. And it was, colloquially referred to as the worm lab. And we went down to the NIH to discuss these worms with other people in the worm groups. And I vividly remember, I don't remember the experimental question, unfortunately, but I vividly remember that a student got up and presented their research, and they had actually quite a large N value or sample size compared to a lot of experiments that we base a lot of our daily life on, unfortunately. Because worms are very easy to grow in the laboratory. They have a very short life cycle. You can look at a lot of them in a few months. So they had a very large sample size, and they had done an experiment and they had drawn a particular conclusion from it. And another researcher stood up and had a very heated argument with his student about how blinded they were to what their data was actually saying because they were looking for a particular outcome, and he thought it said something completely different based on the kinds of outcomes he's looking for in his laboratory and his test. And I honestly thought it was going to come to blows. They were very excited to argue with each other and differ about the interpretation of these results. So keep in mind that when you're setting up an experiment, the way that you set that experiment up can actually, in a certain sense, dictate your outcome. That's one of the limitations of science. You can't find what you're not looking for. There are a lot of things that we're unfortunately not looking for in terms of health outcomes regarding certain medical interventions that were released in 2020, but I'm not going to mention them by name because I don't want to get censored when this goes up on YouTube. So another property of an experiment is that in an experiment, you can only change one thing at a time, and you have to control for everything else. Well, why is that? Well, because otherwise, if you changed multiple things at a time, it would be impossible to tell which variable is actually causing the effect that you're observing. So which change is actually producing that effect, which cause goes with that effect. Now, I like to make a lot of custard. It's one of the few things I can still do in the kitchen, despite my limited ability to walk. And once upon a time, I changed both the sugar and the type of egg that I was putting in the custard. And my custard came out extremely runny. The emulsion broke, and there were little bubbles of liquid in it that should not have been there. And I don't know why that happened, because I changed two things at once. Now, if I had only changed the type of egg I was using, or the egg content, I would have been pretty certain that the change was due to the eggs. And if I had only changed the sugar, then I could be pretty certain that the change was due to the sugar. But then I had to go back and try it all over again, changing one variable at a time to figure out which of those variables was actually causing the effect of the emulsion breaking. And it turns out it might not be either of those variables, because I did everything the same as I used to always do it, and the emulsion broke anyway. So if there's some other variable I'm apparently not controlling for that's causing the same effect. So it's a limitation of experiments. You can only change one thing at a time. And when you control for everything else, you can actually create an environment that is so far removed from reality that it doesn't actually tell you anything meaningful. Now, I'm going to look at a concrete example of this because I don't want to just assert it. And we're going to talk about a long-term E. coli evolution experiment that was done by a gentleman by the name of, I believe his first name is Richard. His last name is definitely Linsky. He started this experiment in February of 1988. So it's been going on for almost 40 years now. And over several decades, the E. coli that he was growing in his laboratory, he claimed that they evolved the ability to metabolize citrate in the presence of oxygen. So citrate is just one possible fuel that bacteria can use. Normally, they metabolize it in the absence of oxygen. So they already possess the ability to metabolize citrate, but he claimed that they evolved the ability to metabolize it in the presence of oxygen. And there was a series of 3 mutational steps to go from not having this ability to having enough of the ability to grow, at a reasonably good clip in the laboratory, although they still don't grow as fast as unmutated bacteria would on glucose, which is their preferred substrate to grow on. It's very simple sugar. In fact, your brain runs on glucose. It's a great thing. So, first of all, when we're talking about evolution experiments, you have to look behind the curtain a little bit and do remember that E. coli here did already possess the ability to metabolize citrate. It just was in the presence or the absence of oxygen versus the presence of oxygen. So nothing new has actually been dramatically done here. But in the first round of mutations, the one that the researchers identified, a graduate student of Linsky's identified, is a very small change to the DNA that damaged a gene that caused the enzymes that were used in the Krebs cycle to be upregulated. That means we have more of those Krebs cycle enzymes, and the Krebs cycle is necessary for the metabolism of citrate. So if I have more of those enzymes, I'm going to get more metabolism of citrate, regardless of what kind of conditions I'm in. So he's kind of setting, it's sort of a priming mutation that allowed you to sort of set up to be using citrate more effectively by upregulating that Krebs cycle. And then there was a duplication of a gene that produced the citrate transporter, and that enabled citrate to come into the bacteria. So when that gene was duplicated, it's really an accidental copy was made of something that was already pre-existing in the bacteria. And it was placed in a place in the genome where it lacked a regulator. So normally, citrate is not taken into the cell unless the conditions are right for its metabolism. So the bacteria normally has all of these sort of protections to make sure it's not wasting energy on doing something like taking in a bunch of citrate when it can't actually use it and eat it. So this was a damage to the regulation of that gene by a duplication being created that placed it out of its normal control sequence and allowed it to be expressed when oxygen was present, whereas normally it would not be present in the cell if oxygen was present. And then that sequence, the third mutation of that sequence, was further duplicated, and that's known as amplification. So now we have more copies of this gene that are running in places that they shouldn't. So now the cell is taking in an awful lot of citrate. And when you add that to the misregulation of the Krebs cycle, then you get the ability to metabolize citrate. Now, at the end of all this breakage of gene regulation, the bacteria still don't grow as well as wild type would on glucose, and they also lost some of their ability to metabolize the glucose. They're actually 20% less efficient at that than the unmutated bacteria. And this is because the citrate metabolism is always on, so it uses up resources that would otherwise be spent metabolizing other more useful sugars. Now, that was hailed as a huge breakthrough experiment that proved that evolution could occur in the lab. But how big really is the deal here? Because we're talking about an environment that's incredibly artificial compared to the real world. Now, in Linsky's laboratory, these bacteria are kept at a constant temperature. They're not subjected to the very early morning chill and the late afternoon heat. They're kept at a very constant moisture. There's no dew fall. There's no drying out. They're kept at a very constant aeration level. So they constantly have the same amount of oxygen, roughly speaking. They always have food available. There's never any time when there's a scarcity of food. And the type of food that's available is always the same. They're not presented with the wide variety of different kinds of metabolites or different kinds of sugars that they would encounter in the wild, in the real life. So all of that gene regulation that got broken to show that these organisms, quote unquote, evolved, that would be necessary in an environment where the temperature was changing and the moisture was changing and the amount of air was changing, the type of food was changing, the amount of food was changing. Bacteria are loaded with these what are called just-in-case genes, genes that you only need in the real world. Now, in the laboratory, Linsky thinks that he got a great evolutionary stride forward in these bacteria, but that's only because he broke a lot of these just-in-case genes. And when he breaks those genes in the laboratory environment, the bacteria will grow faster. But if the bacteria was out in the real world, you wouldn't really see evolution in that sense at all. You would see what you see when you look at this bacteria compared to the wild type on the preferred and most common growing medium, which is actually that they're losing capability and not gaining it. They're losing the ability to metabolize glucose. They're going to grow slower. They're going to get outcompeted by other bacteria that metabolize glucose faster. That mutation is never going to get fixed in the population. You're not going to have any forward mutation. So this great experiment is really hamstrung, and this is recognized by proponents of evolution themselves, not just by people who are opposed to the idea of evolution, that Linsky's basically controlled his experiment right out of reality. That really what he's seeing is an artifact, a laboratory produced artifact, that he would never have even picked up on these mutations if he'd been growing the bacteria in a real world scenario. So that's just one example of the limitations of experimentation. But what about others? So let's turn to some problems with medical research, because this is one of the areas, again, where I have a little bit more expertise. And when you're doing medical research, often you'll find measurements of success as put in terms of significance, so statistical significance. If the p-value is less than 0.05, which means there's a less than 5% chance that this could have happened by chance, then usually that's reported as a significant result. In fact, I think always it's reported as a significant result. Now, just playing statistics here, if you look at 20 outcomes, odds are that one of them is at least is going to be positive. One of them at least is going to turn in the way that you want it to turn. So if you're looking at a drug and you're looking at its effect on the human body, you can measure a bunch of different things. You can measure, if you're looking at something that affects respiration, you can look at oxygen saturation, you can look at respiration rate, you can look at resting basal metabolism. There's all different kinds of things you can look at. And if you look at 20 different outcomes, odds are one of them is going to be statistically significant. And then you report that one. And that has actually been done before in clinical trials. It's documented in the book Bad Pharma by Ben Goldacre. And another problem with medical research is that you'll sometimes get researchers who will use proxy measures. So when you're looking at an outcome of an intervention, so we'll take for a specific example the HPV vaccine. The HPV vaccine is supposed to prevent cancer, and cervical cancer in particular. It's supposed to then, by extension, extend your life and reduce your risk of death from cervical cancer in particular. But when they trialed this vaccine in real life, they didn't actually measure whether the participants who received it or didn't receive it developed cervical cancer. They measured cervical lesions, and these lesions occur when you're infected with the HPV virus, but they will often resolve on their own. The particular grade of lesion that they looked at often goes away by itself. It doesn't necessarily lead to cervical cancer. So when they looked at the efficacy of this vaccine, they measured whether these cervical lesions went away. A lot of times they'll go away on their own anyway, and they use this proxy measure to say that the vaccine was effective, but they didn't actually measure whether there was a decrease in the incidence of cancer, and they didn't actually measure whether there was an increase in life expectancy, which are the two outcomes that they're aiming for. And this is a big deal with this particular intervention, because it turns out that the rate of HPV in the population hasn't really been affected by vaccination at all. Instead, what you've seen is strain replacement. So the strains that are not in the vaccine are now more commonly associated with cancer, and they're the ones that produce the most cases of cancer. So then we had to, not we, but the manufacturer had to create a new vaccine that had more strains in it, but we're still just seeing the strain replacement problem because we didn't actually see if this vaccine prevented the real endpoint. We only saw if it prevented the proxy measure. So that's one problem that you have in medical research. Another big problem you have kind of across the board in research is attempting to quantify something that is unquantifiable. So we'll talk a little bit about materialism and how materialism kind of leads us to the idea that since there's only matter, then if you can quantify it, it matters, and if you can't, it doesn't, because matter is that which has mass and takes up space, and those things are quantifiable. So what do I mean by quantifying something as unquantifiable? Well, let's take the example of a pain scale. I get asked a lot what's to rate my pain on a scale of 1 to 10. So think about that. Think about your day in the last 24 hours. What would you say is your highest level of pain on a scale of 1 to 10? Well, what's 10? And I mean, 0 I get. 0 is nothing. But what's 10? 10 can be subjective. The most intense pain I ever experienced in my life was falling down and breaking my ankle. It really was tearing a tendon in my ankle, but it will not quibble. I haven't experienced any pain like that in a really long time, but I've experienced enough pain that I'm pretty incapacitated. In fact, it's taken me 3 months to record a presentation. So what is that on a scale of 1 to 10? I don't know. You tell me. Now, if you try to compare that across multiple people, so you take everyone in the world who's torn their tendon in the exact same way that I did, and you ask them to rate their pain they're all going to have different measures because they all have different ideas of what is a 10 and what is a 5. These things are not actually quantifiable, but when we put numbers on them, we reduce them to something that can be measured in that way, but it leaves a lot of nuance of the reality out of it. Again, you're controlling your experiment right out of reality. Now, another problem with medical studies in particular, this doesn't, it sort of translates into regular studies because you can just, dismiss data points that don't quite fit. But when people in medical studies drop out, that data gets dismissed. Those people are just considered lost to follow-up. They're not actually counted in terms of efficacy or in terms of adverse events. So I recently just met someone whose husband dropped out of a cancer study because it made him feel so unbelievably sick. Well, when they report adverse events that are associated with this medication, they're not going to report what he reported because he's lost follow-up. He doesn't count in the data anymore. That's one way that the data can be manipulated. And in fact, all these problems we're looking at in medical research can lead you to draw conclusions that are positive or negative when you shouldn't be drawing a conclusion at all. You might be drawing a conclusion that there is significance when there isn't. You might be drawing a conclusion that there's a positive effect when there isn't, and even worse, you might be drawing a conclusion there's a positive effect when overall there's a net negative effect. So the stuff matters a lot. It's not just semantics that we're arguing about right now. So sometimes people drop out. Sometimes only a subset of data is examined. There was a Pfizer study on a certain medical intervention that was released in December of 2020, in which there were about 40,000 participants, and the data that was analyzed in order to determine the efficacy of that intervention relied on a few hundred people out of that 40,000. That is less than 1% of the overall people that participated in the study that were used to calculate the efficacy. So if you wonder why that particular intervention didn't prove particularly efficacious in the long run, now you know. And again, I'm not naming it in particular, but I'm sure you all know what I'm talking about. Sometimes in medical studies and in other studies, data can get bundled in weird ways. So when you're looking at statistics, you're losing some level of nuance. You are not, when you're quantifying things, you're not talking about the qualitative aspects of them. You're not talking about how one thing might differ slightly from another. So in Pfizer's RSV vaccine study, the data regarding adverse events was bundled, and some things with the digestive system were bundled together. And in the same category were things like flatulence, which is not particularly harmful, and inflammation that damages the esophagus, and also umbilical hernias. Those things don't go together in terms of their gravity. They simply go together in terms of their body system. So when adverse events are bundled in medical research, you can sometimes hide that there is more adverse events in the group receiving the new medicine than there is in the group that's receiving the control treatment. And in that study, one particularly crazy result was that there were 27 babies in the 4 experimental groups that were jaundiced, and only 1 baby in the control group experienced jaundice. Now, jaundice is an indication that something's going on not quite right with the liver, and that's a pretty important organ. So I would think that you would report that instead of bundling those results so that it looked like, that there was no difference between the experimental groups and the control group. And ironically, the only baby in the study who got RSV was in the experimental group receiving the experimental vaccine. So another problem that you can get in research, medical research and regular research, is publication bias. So papers or projects that show no result, that show that there is no relationship between your factors, that there is no effectiveness to the intervention, or that we can't tell if there's any effectiveness to the intervention, they don't generally get published. Papers that show poor results for the people who are funding the project also don't generally get published. It's really important to remember scientists are not strictly objective. They are beholden people. They're beholden to their peers who kind of control whether they get published or not through peer review. They're beholden to the federal government if their funding is coming through the NIH or through some other government agency. They're beholden to pharmaceutical companies or military industrial companies if their funding is coming from a private source. And all of that, reminds me of a meme that I saw recently that said, I try to follow the science, but it keeps leading me back to the money. Publication bias is affected by funding as well. And, if you don't publish, you don't get funded. So you have this whole publish or perish phenomenon that we'll come back to here in a minute. But the last problem I wanted to talk about in medical research, and it's not the last problem, it's just the last one I have time for today, is outright fraud. And again, I'll use a particular example rather than just claiming that there's fraud in medical research. And this particular example is a researcher by the name of Sylvan Lisney. Published a paper on Alzheimer's disease in 2006. And this paper is the one that established really that the beta amyloid plaques are the main cause of Alzheimer's. These are the things that the target of basically every Alzheimer's drug under development. They're the target of most therapies for Alzheimer's. This paper was a big deal. It became a foundational uh, portion of Alzheimer's research. Now, in 2022, a whistleblower, another PhD researcher, noticed that in Lesnay's paper, the for those who are technically minded, the bands on the Western blot were the same shape. They were just shaded out to be slightly different. So basically what he did for the less technically involved is he took a piece of a photograph of his research results, and he cut and pasted it. But before he did that, he kind of lightened the color up a little bit so it wouldn't be as immediately identifiable as that he'd cut and pasted it, but he did actually cut and paste it. And this kind of fraud was detected in a number of his other papers. And this case started, it hit Science magazine in 2022, and the papers weren't retracted until 2024. So Science moves very slow at self-correcting, and in fact, often it doesn't, which is really my next point about one of the limits of experimentation, which is that science is not really replicable, and it's not really self-correcting. In the publish or perish environment, where scientists really need to actually get publications in order to keep their jobs or in order to keep their funding, you basically have a situation where you end up with a lot of publications that are basically like pointless. For example, the British Medical Journal in 2017 did a review of cancer drugs, and they showed that in the studies that they looked at, the magnitude of the benefit on overall survival ranged from 1 to 5.8 months. So these drugs that are being published as really good and candidates to move forward on and start clinical trials on are buying cancer patients an average of 3 months. That's it. That's really kind of a horrifying statistic when you consider the side effects of cancer drugs. So we have too many scientists spending too much money on research, which I say because there were 8 million papers published in 2023. 8 million papers. That's more than anyone could possibly read in their lifetime. And you think about the number of papers that were published on a particular virus that started circulating in 2019, there's so much out there you could never read it all. And a number of them were never peer-reviewed, a number of them were retracted, and the important ones just kind of get lost in the sauce because there's so many papers. So really what we need is actually probably less STEM education and less NIH funding. Although I don't think there are too many people out there advocating that, unfortunately. But this drive to publish really it not only results in just basically banal publications, but it really kind of it causes an unwillingness to challenge the status quo. And I saw that myself firsthand when I was working in Dr. Venegala Rao's lab. And I said, well, we're here at the Catholic University of America, which is the Bishop's University, in the United States. It's the Catholic university. And we're doing research using abortive fetal cells. Maybe we should do something different. And he basically argued that, well, this is what's in the literature. This is what other people have used. Therefore, we have to use it because we have grant funding on the line. We'd received the first part of a grant from the Bill and Melinda Gates Foundation, and we were working towards receiving the second part of that grant. And if we didn't do it fast enough, we weren't going to get our grant money. And so why would you change? Why would you do something different? Why would you reinvent the wheel? And I think a lot of people who don't work directly in science are not aware that there's so many incentives, institutional and financial, to really just not challenge that status quo. I have to get published to keep my job. I don't want to upset the peer reviewers. I don't want to do something different. I don't want to start at square one. I want to build on what other people have done. And I'm assuming that they're correct. But there's pretty good evidence at this point that a lot of these papers that are being published are not. And there's some documentation about replication basically not happening or failing, that was put out by John Ioannidis. And he looked at 49 papers that had over 1,000 citations each. So these are some of the most cited papers in his field. And he found that about 16% were just flatly contradicted by later studies. Another 16% had effects that were stronger than were reported in later studies. So they were reporting that their effects were more significant or more meaningful than they actually were. So this, just to use an example on the cancer patient, this would be somebody reporting that their drug, increased life expectancy by 12 months when really it only increased by 6 or 3 or something like that. About 45% were actually replicated, but another quarter of them simply were not. No one ever looked at it. No one ever tried. So you have to ask the question: why are a quarter of these papers completely unchallenged? And really, being a whistleblower in this environment is really, really tough to do. It makes you a target for defamation lawsuits. There are lawsuits going on right now. In a really big scandal in a business department. I believe it's Harvard, but I don't remember exactly the school. But there was a researcher whose work was identified as fraudulent, and now she's suing the person who identified it and the school for defamation, because it's obviously going to ruin her career if she's faked it. And it's actually pretty clear that she has, and those charges, some of those charges did get dropped. But when people have money and have power, and we live in a litigious society, it's frightening to come out and not agree with your peers, or to point out that actually I think your work is questionable. And it's an unfortunate part of this whole system, which becomes self-referring, that junior scientists are just very unlikely to challenge senior scientists who have tenure, because they don't, and their jobs are going to be at risk. And so this kind of leads me to my next point, which is that scientists are not perfectly reasonable people. We have this image in our cultural mindset that scientists, when you put on that white lab coat, you put on some sort of purity and some sort of righteousness of intention. And you can't lie, and you can't be deceived, and you can't be motivated by anything other than the pursuit of truth. And you can't make mistakes. And so to just kind of upend further that illusion, I'd like to give a concrete example of a pretty big mistake that happened in recent memory, in the lifetime of a number of you. So in the 1960s, Soviet scientists were experimenting with condensing water vapor in tubes that were less than 1 millimeter in diameter. And they found that the water behaved very differently in these tubes. It was denser and more viscous than regular water. It had a different freezing and melting point. It turned into a weird glass-like substance at minus 34 degrees Celsius. And they said that there was no contamination of the water, so the water was clean, and there's no contamination of the vessel. So they concluded that under these circumstances, water takes on a new form. And this was validated by the fact that this water, this polywater as they called it, didn't match any substance in their absorption spectrum database. So they assumed it had to be a new thing. And this research was replicated. There were over 100 papers in 1970 alone that were dealing with polywater and working with polywater in the lab. In fact, somebody isolated almost a gram of polywater, which is about the weight of a paperclip, but it was considered to be a lot at the time for this new substance. In 1971, it turned out that polywater was actually human sweat, and that apparently there was contamination of the tubes, and it had the exact same chemical composition as sweat, and it behaved exactly like sweat, and it wasn't new at all. But for a decade, people were looking for polywater and for this new behavior of water. This isn't the only time that people have flubbed with water. And I actually would refer you to there's an interesting SciShow episode on this, about what happens when scientists are wrong. And I won't go into the whole story, but some of you may remember that they were looking for canals that they presumed were built by intelligent beings on Mars right up until 1964. Now, these canals were first envisioned by a gentleman looking through a telescope, and it turns out that when you stare at something for a really long time, your wonderful human mind that searches for meaning and understanding in everything starts to connect things that are not necessarily connected. So he spent so much time staring through his telescope that he kept identifying canals and new ones all the time. So they assumed they were being built. And other scientists were looking for them right up until 1964 when they realized it was really just an artifact of this particular scientist's imagination and the time that he spent staring through the telescopes. So scientists can get tired. Scientists sweat. Scientists can make mistakes. Scientists are not some perfect, reasonable arbiter of truth simply because they put on a white lab coat. So that finally brings us to scientism. And I'll use William Brigg's definition because I love it. And he says loosely, scientism is the proposition that science has the answer to all the questions, if not now, then in the future. So scientism is tied up with a lot of other isms. This idea that science can answer all the questions is tied up with naturalism, the idea that the world can be explained solely through natural laws and natural forces. There's nothing outside of the laws of nature, which is, very similar to materialism. The idea that only matter exists. There's also reductionism, the idea that all life and really everything that exists is ultimately reducible to physics, to the interaction of the atoms of which this matter is composed. There's determinism, the idea that every phenomenon, including our own actions, is determined by a pre-existing cause, which is itself determined by a pre-existing cause, which is itself determined by a pre-existing cause, all the way back to the Big Bang. So the nothing that the universe is supposed to have come out of, which it gives physicists really a run for their money. If you look at the way that physicists try to define nothing, they can't quite seem to do it, which is an interesting problem in physics. But the way that nothing was arranged somehow determines the fact that I'm sitting here giving this presentation several months late. That's determinism. And then, of course, neo-Darwinism, which is my least favorite, which is the application of the theory of natural selection to all of human behavior. This idea that the fact that I can communicate with you somehow had to have evolved through natural selection. And I'll quote Andrew Ferguson because I like what he has to say about this. He says, the conscious brain that is able to come up with neo-Darwinism as a universal explanation simultaneously makes neo-Darwinism as a universal explanation exceedingly unlikely. The fact that you can think about the fact that you could have evolved from slime suggests that something other than slime was involved in you getting here. And really, the idea that man came from a warm little pond violates a lot of first principles of philosophy. And I don't have time to talk about them here, but I'll just refer you to Father Ripperger's excellent book, The Metaphysics of Evolution. It's a short book. It's a dense book. But it talks about a lot of the philosophical problems with Darwinism and neo-Darwinism. So all these things tend to be meshed together. This idea that science has all the answers, because of course, science works through empirical methods, works through experimentation, like we said before, works through testable hypotheses, and looking at matter, and matter is measurable. And so, if science is the final arbiter of truth, if science has the answer to everything that ever will be asked, if not now, then later, eventually, and when we come up with better technological developments, then of course, all these things are going to be involved in materialism, the reductionism, the determinism, and in our cultural climate, the neo-Darwinism. So keep in mind as we go through this that materialism really is synonymous with liberation from religion. Scientism is popular, I think, primarily because those who choose atheism have to have an explanation for everything. And if they have an explanation for everything, they can reject religion. They have an explanation for how I got here, they have an explanation for where I'm going, and they have an explanation for everything in between, or they think they do. And that really should serve as a reminder that the greatest crime in our age is not the things they're doing to kids in hospitals, which again would get me banned from YouTube, whether before they're born or after they're born. It's actually blasphemy and atheism, because these are the things that lead us to accept the idea that gender is a continuum, or it's my body, so it's my choice. But to return to our question, can science explain everything? Well, I'll reply with a few questions of my own. I have a minivan, which is on its last tire. It's absolutely completely run down and kind of a bucket of bolts. So, should I buy a new car? Or should I buy a used one? Or can I make do without a car? Can science answer that question? Well, there are some things that I can measure and tabulate that might give me some information that would help me answer that question, but science can't really tell me how I should spend my money. What about this one? Should I go to college? If so, what should I study? Well, science might say science, but science can't arbitrate life decisions for you. Science can't tell you what vocation you should choose. And to escalate it even further, what about the question, what does it mean to be loved? What does it mean to be loved by my parents, or by my friends, by my spouse, by God? Science doesn't have the answer to any of these questions. And it turns out that materialism is an explanation for a world we just don't live in. So I'm going to borrow and expand a little bit here on an argument from Edward Fazer, because he's a philosopher and I'm not. And he argues, and I think he's correct, that scientism is not a coherent position. The idea that science has the answer to everything can't be maintained. Because we just saw from the questions we were just asking that science doesn't have the answer for everything that matters in life. And even if you want to argue that these things I was asking you about don't actually matter, you're still stepping outside the bounds of science. You're stepping outside the bounds of experimentation and hypothesis and falsifiability. So you can't hold that experimentation is the best or only way of knowing something without stepping outside of science. You have to make an extra scientific judgment. And here you are trying to avoid having extra scientific judgments, but you can't, because the very attempt to do so involves having extra scientific judgments. And if you think these judgments are rationally justifiable, so if you think that scientism is true, you've de facto admitted that there are positions outside the purview of science that are rationally justifiable, because science can't tell you that scientism is true. Science can't tell you anything about worldviews. And therefore, scientism commits what Greg Koukl, from the author of Tactics, calls formal suicide. The idea kills itself in its own wording. You can't maintain that science has the answer for everything that matters in life when science can't even tell you what matters. And again, Dr. Fazer, for those of you who are more philosophically inclined, put that argument in much more technical terms in his article, Fallacies Physicists Fall For. So I'll refer you to that, and I'll try to put a link in the video notes here so you can get to it directly. Fazer also pointed out that there's a problem that comes in when you point this out to people, because the fallacy is actually so blindingly simple. It's so easy that my best friend's 5-year-old could see it. When you point this out, it can be a huge blow to the pride of the person holding the fallacy. And their response often is, well, surely you must be wrong. Like, you must be thinking about it incorrectly. But it doesn't matter how confidently they say that. That's not evidence. It's just an assertion. And don't let them fool you. Don't let people who are arguing with you fool you. Don't bow to a demand for an operational definition of a philosophical concept. That is, for a concrete, replicable procedure, experimental procedure, designed to represent that concept. You can't give them that, because science can't do philosophy. And don't try to give them experimental evidence for it either. Just point out that they're begging the question. They're assuming that science is the only arbiter of truth, and therefore you have to give them an operational definition or experimental evidence before they'll believe that something that you said is true. So that's kind of up here intellectually. So let's make it concrete. And I'll point out that it's concrete by telling you that testing a hypothesis presupposes that there are things that are not testable by science that are true and reasonable. Hear me out. We know that change occurs. We know from our experience change occurs. I don't have quite as much hair as I did when I was 20. It seems to be thinning. Maybe there's more lines on my face, more wrinkles. Things change over time. A baby is born, they grow, they reach adulthood, and they get old. We know change happens. In fact, to say that change doesn't happen is kind of also philosophically untenable. But for a whole argument about that, you can see the first couple of chapters of Dr. Fazer's book, The Last Superstition, which I have over here on my side table. So we know that change occurs, but the fact that change occurs is not testable because it's not falsifiable. I can't prove that change doesn't occur. I can't even design an experiment that would demonstrate that change doesn't occur because the very possibility of falsifiability presupposes change. You predict what you think will happen, you see if it happens, and then you go from wondering whether it will happen to knowing yes it will or no it won't. So the very fact that you can test a hypothesis presupposes that there's something that's not testable by science. In this case, that's change. And that's just one example. Again, Fazer has many, many, many more. So to sum up the problems with scientism in a nutshell, because I'm coming up here closer to my hour time limit, although no one's giving a talk after me, so I could keep going technically. I'll quote Andrew Ferguson from his excellent article on Thomas Nagel and how he's eschewing strict materialism. And he says, a materialist who lived his life according to his professed convictions, understanding himself to have no moral agency, because of course, and this is my aside, after all, everything goes back to how that nothing was arranged during the Big Bang, which caused something, which caused something else, which caused your actions, which is determinism. So he's understanding himself to have no moral agency, seeing his friends and enemies and family as genetically determined robots, because, again, my aside, after all, as Francis Crick said, your joys and your sorrows, your memories and your ambitions, your sense of personal identity and free will, are in fact no more than the behavior of a vast assembly of nerve cells and their associated molecules. Who you are is nothing but a pack of neurons. This is a preeminent scientist giving his opinion of materialism. So to go back to the whole quote, in case I lost you there in the middle, Andrew Ferguson, a materialist who lived his life according to his professed convictions, understanding himself to have no moral agency, seeing his friends and enemies and family as genetically determined robots, wouldn't be just a materialist, he'd be a psychopath. And so when you meet scientists, they behave as though things outside science exist. They behave as though there's good and bad, and that you're a good person if you do the good things and you're not if you don't. And you have to try to be good and not to be bad. And, no one argues that, it's perfectly acceptable to punch them in the face. But if you were a materialist, well, the Big Bang was, the molecules were arranged and something happened that caused something else. And that's why I punched you in the face. It's not my fault. But nobody lives that way. So the final question is, at least the final question we'll address here, is if scientism is such a problem, if it's so philosophically unsound, and if it leads to such terrible conclusions, if you actually carried it out to its full extent, how did we get here? How did we get to a point where we can deny first principles of philosophy and claim that we're actually still having rational argument? And how do we get out? So if I didn't mention it too much before, but I have been a teacher for a number of years. I've taught science, again, from middle school to elementary school all the way up to college. And I think a lot of the way that we got to the position that we hold now about scientism goes back to two fundamental errors in the way that science has been taught in schools, at least since I was a kid, which has been about 4 decades now. But I'm sure it's been taught that way longer. And the first error that occurs in science education is an inversion in the order of knowledge. So we literally flip the way we learn upside down. And because true knowledge starts with what we truly know, what we experience, and works our way out from there. We don't start with a Cartesian doubt. I think, therefore I am. Because everything else I can doubt. We, as babies, we put everything in our mouth to see what it is, how it tastes. Our study of everything really starts with our senses. It starts with our intake of the world around us. We see something and that informs our intellect. Through our sight, we hear something and we start to recognize it as a pattern. We begin to learn language. We start there with things that we actually know, with things we actually experience, because it is true that all knowledge comes through our senses. And then when you invert the order of knowledge, as they do in science curricula currently, you don't start teaching children about things that they know, and then leading them to the next step. And this is actually what we do in our biology book, Biology: A Traditional Catholic Perspective, which is available on the Kolbe Center, Kolbe Center's website, the Kolbe, Center.org. We flipped the curriculum in biology. Normally it starts with molecules and biochemistry. We started with your senses and helping kids understand how their senses work, how hearing works, how sight works, because this is how your information is coming in. And then we go to the human body, because they're living in a human body. They have an understanding of a human body. They know what a human body is in a way that they don't know what a plant is. And then we work down to animals, which are more analogous to humans, and then plants, and then molecules. Because each step along the way, they're learning something more that they didn't know before, but they're always hanging it on what they had before. Now, the way science is usually taught in schools is actually backwards. They start with a molecule and teach up to the man, and you usually will get anatomy and physiology at the very end if you get it at all. And this takes a concept like an atom, which is almost mythical to students if that's where you start, and places it as the building block on which you're going to build this entire edifice of chemistry or biology. And so they start out from the very beginning of their education in science, from the first moment, having to take the scientist's word for it because they don't understand an atom. And here's where I kind of need to work in the concept of models. And if you only remember one thing from my presentation, well, first remember that scientists are real people and they have bad days and they have motivations just like you. But second, remember that models are really important in the way that science is currently taught, because a lot of science that is being taught in this inverted order, where you start with something the kid is completely unfamiliar with. So you have to have a model, because children, especially in the elementary age, they're very concrete. And if they don't have something to look at, they can't really conceptualize something abstract. So what we end up with when we teach science the way we do is that representations of reality, these models, are often mistaken for reality. So for example, if I ask you to picture an atom, what do you see in your head? It's probably something with a little tight ball in the middle and some loopy things around the outside. Whether it's that traditional sort of six-pointed set of 3 loops, or just some general loops. You're picturing something in your head. But you're not picturing an atom. You're picturing a model of an atom, a model that's been presented to you at some point in your education in science. It's something concrete that we can grasp with our intellect and generally with our vision to explain something that's extremely indirect and seemingly unrelated in terms of the observations that we're making. So, for example, when you picture an atom, nobody ever pictures Rutherford's gold foil experiment. You picture the model that's based on that data, and that data is very, very indirect. So the way that we present science leads us to this confusion of model with reality because we've inverted it. We don't have anything to hang our understanding on. We look at pictures of reality. We read words about reality, for those that do actually read the textbooks, and we don't really interact with reality. And there's kind of an additional source of error that goes along with this inversion. And it's that students are often taught things before they're conceptually able to understand them. So I used to judge science fairs, and I would judge elementary science fairs. And first and second and third graders really have no concept of the scientific method. They can kind of be walked through, like putting things in the right boxes by their teachers or their parents. And if you ask them, they might tell you, yes, this is the independent variable. But by the time I would get them and teach them in 10th or 11th grade, I would have kids who didn't understand the difference between a constant and a control, because those two things both start with C and they have roughly the same number of letters, and they were just memorizing things. They didn't understand that there are things that you have to keep the same in the experiment and you can't change, because otherwise you don't know what whether your variable you're actually testing is having the effect that you're actually seeing. And they don't understand that you have to run a whole separate simulation where you don't introduce any change that you're trying to observe at all, and you see what would have happened if you'd done nothing. Because if you don't have that, you can't really say that the change was due to something that you did. My custard example I had earlier, I had no control. I didn't have a custard that was made the exact same way as I'd always made it before, baking alongside the custard that had the new sugar and the new kind of egg. So I didn't know whether one of the things I changed or something that I didn't realize was causing that effect that I was seeing. So we're trying to teach students the scientific method before they're really able to reason abstractly in the way they would need to use it properly. And therefore, they come to me in 10th grade and they say, "Our hypothesis is an educated guess, and I don't understand what a control is." So how do we fix this? Well, one of the things I think we should do is keep kids away from textbooks as long as possible, especially in science, and really allow them to interact with the real world, to learn observational skills. And I could go on about this for an entire other presentation. In fact, I have at some homeschool conferences earlier this year and our annual Kolbe Center leadership retreat in August. And that talk was recorded and will hopefully be published to YouTube sometime in the near future, at least hopefully before our next conference next summer. And we also actually, I wrote up a series of recommendations that's available on our website under the title Wonderful Are Thy Works. And it's a whole explanation of these problems with scientism and education, and also a whole set of recommendations on what to do about them. So I won't go too much into that here. But I do have to address the second error in science education, which is really unconditional faith in the science. And students are groomed in this, even in the way that popular representation of scientists are presented. So not just with the models, not just with having to take the teacher's word for it or the book's word for it, but if you look at children's books on Einstein, on Edison, on Newton, they just seem like larger than life heroes. They kind of never did anything wrong. They had all these fortuitous things happen to them, and they really, the way that they triumphed over everything was rigorously following the scientific method. You will never see in a child's book on Newton that Newton was an alchemist. And don't take my word for that. You can go look it up. That's a problem when it comes to his interpretation of his results and his worldview. And that's also very much outside of science. So the remedy for this second error, I think, is that students need to develop a healthy understanding of both the power and the limitation of experimentation as a way of knowing. Because there are a lot of things that we can learn very well from experimentation, but they're only related to things that we can measure, things that are material and things that can be quantified. So the best way to do this, I think, is to do an actual experiment. And this is where I would actually recommend science fairs, just not until kids are much older and can actually design their experiment without a whole lot of help and input. Because a student, if you do a single reasonably well-planned experiment, it doesn't have to be perfect. It doesn't have to be great. They'll learn one of the most important things they could ever learn, which is that data do not come gift wrapped with a conclusion. Experiments are messy. Data is messy. There's lots of ways to screw it up. There's lots of ways to interpret your data. As we saw earlier with that set of data that was given to 71 groups of scientists and they came up with 1,200 different models. It's not as simple and straightforward as putting on your lab coat, going into work, and looking under a microscope and seeing truth. That's not how it works. So I have talked for a little over an hour, so I'm going to let you go, return to your lives as normal. But just to sum up, we've talked a little bit about the nature of experimentation. We've talked about its removal from reality, its reliance on falsification, which further relies on the reality of change, which is itself not falsifiable. So experimentation is a limited way to know truth. We've talked about some particular problems with medical research, including using proxy measures, reporting trivial results, and even outright fraud. We've talked about how scientists are not perfectly reasonable beings, but humans, just like you and me. And we've defined scientism, basically science having the answer to everything, and explained why it's self-refuting from a philosophical standpoint. And again, for further research, I really refer you to Edward Fazer. He does a lot better job explaining that in technical language than I do. And we've discussed a little bit about how we as a society got here to where we can commit some of these philosophical flaws, which you can dive more into in some of my other presentations if you are so inclined, or in that book, Wonderful Are Thy Works. So now I hope I've just left you with a little bit better understanding of why friends don't let friends trust the science. And if you're interested in more work like this, please go to the Kolbe Center's website. It's, again, KOLBE center.org. And there's a lot of stuff on there that's very related to these topics, particularly on the subject of evolution, but there's also some health things. And of course, you can check out some of my other talks on YouTube here on this channel or on Census Fidelium, which is run by my friend Steve Cunningham. Yeah, I hope that God blesses you and keeps you well. And thank you for your time and listening to this. I really, really appreciate it. So let's close with a prayer. In the name of the Father, and of the Son, and of the Holy Ghost. Amen. Glory be to the Father, and to the Son, and to the Holy Ghost, as it was in the beginning, is now, and ever shall be, world without end. Amen. God bless you all.